So my name is Mary Wang. I'm the biobank program manager at Fondazione Teleton in Milan, in Italy. And also with RTConnect, I'm the project manager for coordination of biobank work package and tool development, and also part of the coordination for your biobank. With me today presenting this webinar is David van Enkevoort. He's a project manager at the University Medical Center of Groningen in the Genomics Coordination Center in the Netherlands. And so in RTConnect, he's also in the work package developing biobank IT tools. And he's also responsible in several biobanking projects, also in the BBMRI NL and BBMRI ERIC for biobank directories. So he's our biobank IT expert. So I'll make us start now. If you have any problems for hearing, or if you don't hear me well, if there are noises, please let me know. Or I think you can unmute yourself, or you can also write a text chat to let me know if you have particular questions. Okay, so I'm going to start off the webinar with an introduction on the work that's been done by RTConnect so far. And this is very much a joint effort as a European program. And in the biobanks where I work with Lucia Monaco, and there's several tools being developed by many people in Europe. So registries and biobank finder with the team in Austria, and David in the team of Sample Catalogue, and many others, and many of you, some of you actually also been very much involved in these years in development of the tools. This webinar is the second run of the webinar designed for biobank managers. And this is the dedicated design for you, especially biobank managers for in the Euro Biobank network. And just now we have 25 rare disease biobanks scattered in 11 countries. So we're looking forward for you to get more and more involved. Hello?
Hello?
Hello, you have a question?
No. I'm just testing if the connection is okay.
Yes. Okay. Yes, I can hear you.
Oh, thank you. Whom?
You? Okay. I will continue. So now RTConnect, page down, has developed now several flagship tools for rare disease research. There are three major flagship tools. So the Sample Catalogue, which we'll look at in more detail today. The registries and biobank finder, a directory of rare disease registries and biobanks, and the genome phenome analysis platform allowing researchers to do genome phenome analysis directly online. So all these tools have now a lot of resources, and the numbers are increasing. In the Sample Catalogue right now, there are just over 7,000 biological samples, and this webinar is dedicated. So now with the aim to increase this number, and so that researchers can look at and find samples of that they need for their research, and make your sample collections available and accessible to the other researchers. There's a little difference between the registries, biobank finder, and the sample catalog. So the finder, as I said, is more like a directory. So it's a collection of information on the biobank and the registry itself, such as the address, the contact person, what kind of SOPs it follows, and as such. You can look for a registry or biobank via disease name or min. And the sample catalog instead is really on the samples of the biobank. So, DNAs, which diseases, male and female, really much more detailed information about the biological samples. And you can look up information. Researchers can look up information via disease name, different codes, and have various filters to refine their research for biological samples. This is the web link for the RD-Connect sample catalog. So it's samples.rd-connect.eu. And now I just actually want to run through a quick use case to show you what researchers can do with this platform, with this tool. So just example. So if I want to find 10 DNA samples from females with ring chromosome 14 syndrome, how I would go about in looking for that in the sample catalog. So when you go in, you see a grid with lots of names, but so you usually will start with the search function. And so here you can type in disease name, such as ring chromosome 14, or using the orphan number. After that, you'll see that the disease is already refined to what you searched in. And you can see there are some real material types, male and female, and such. So what you would do is you would try to refine their search again using the filters. So in our example, we're looking for DNA samples in particular and from females. So you could search, filter for females and DNA. And there, at the end, so they keep refining, and you here in this example they show, so the researcher would have found, I found 14 DNAs. So that fulfills my search criteria. You can also search just to want to know, you don't have a specific numbers here, but you want to know which biobanks are holding the samples. So in here, which biobanks are holding plasma samples for this dystrophy. And so you can search in a similar way and doing different filters. And here you can see that there are 2 biobanks holding plasma samples for this disease. And later on, right now, researchers, the catalog is open for all users to browse. And later on this year, we are implementing also a tool to allow users to also make requests. So at the moment, people can browse and see where the samples are, who they should contact. But and soon this year, we're implementing it so that once they have found the samples that they're interested in, they can make the request directly and reach the biobanks more efficiently. So now I'm going to pass my screen sharing to David, who's going to show you how, as a biobank, how you would be able to share your sample catalog to the RD-Connect platform so that researchers can look for the samples in your biobanks. David, I've stopped sharing my screen.
Okay, thank you. You can hear me well?
Yes.
Thank you. So to get the data from your biobank into the catalog, we can use our upload. So that's first part. I'm going to explain that. So I can upload Excel or CSV files. So this is basically the process. You extract the data from your system and then send the file to upload in the Sample Catalogue, and it must be encoded to the structure that is the same if we want to be able to integrate all the different biobanks and search across them. So the process is we start out with your actual sample list. So in the way that you have it in your system, and describe the structure of your sample list, and then upload that into the catalog. So the items that we need are the following. Important here to note is that identifiers should be pseudonymized, and all items that are marked with the star are the required fields. So you could have just an Excel file like this. Then we described the structure of that in a way that that the system can interpret it, where each of the sheets in an Excel is an entity or a table in the catalog, and a column in the Excel is an attribute, and for each of them we have to describe if what kind of data is in there, and if it is required to be there or not. Then in the catalog, if you are administrator for biobank, you can use admin menu upload to upload the data. So you select your file that has been prepared and click on next, and then first time upload will add the entities, and if you later on do another upload, then you probably want to add entities but also update existing entities. But if you only are adding updating items, then you might also want to maybe select just update. The system will then start validating your data. In the second part of that, you get this report that will say if your data is importable, and also for each of your sheets in Excel, if there are any required fields missing, or unknown fields or optional fields that you didn't fill in. So this is an important step in the upload process to validate, and in the end it will report how many items have been uploaded, and if you and then it can be shared and can press finish when you're done. So then the result is that you can see your data in our data explorer. So in there you can search for your data set, and then you can see the data and see that if it's imported correctly, and also you can still edit the data, so you have forms to change some of your data. But if we just upload your data in the format that you have, then we still do not have a common standard. So the other part is how do we convert data to adhere to the same standard, to make it comparable. So as a reminder, in the end, we would like to have these fields that we agreed upon in the user panel for the catalog. So star again are required. Some of these, like material type, diagnosis, etc., have specific acceptable values, as you can see here. So notable there is that disease is a term from the ORPHANET ontology. So if your data would be exactly in this format already, then great. We don't need to do a conversion, or if you can deliver it in that format yourself, okay. So you also have upload template for the catalog data format available on the website. But if your data is not in this form, then we have in our system our mapping and recoding service that can help to convert your data to the format of the catalog. So this is something that at least in the coming period till the end of the financing of the ID Connect project, we can help you with our data managers to create a mapping of your data. So the first step is that we go to the mapping service and create a mapping project. In the mapping project, we say we give it a name, and we say that we want to map it to the ID Connect sample catalog entity. So the data model with the attributes with the fields that I showed before. Then we get to this screen where we can add a source. So that is the data from your biobank as it's been uploaded in the original format. So here we can select one. and then the system will try to best guess the actual mappings, and it will encode the mappings with red for things that have to be discussed, but yellow, blue are for the guessed mappings, where the color gives blue is a higher confidence, and green is anything that has been validated by you as biobank or our data managers. So for each of these mappings, we can click on the pencil icon to edit them, and then get into this screen. This might be a bit intimidating, but I'll explain it in more detail. At the top, there is some information about the field that this mapping is dealing with. If it is a list of acceptable values, then that list will be there. The name of the field is there. Then at the bottom part are 3 panels for the attributes from your set, how they can be converted, and results of testing. So looking into that, the system will show potential mapping attributes from your data, but you can also search in the data, and you select the one that you one or multiple that you need for the mapping. Then the middle panel is allows us to create a small script to convert your data into the RD Connect data model. If it is just the same value, then it can be a really simple script, but if necessary, you can do also more complex mappings of, in this case, words into ontology codes for the mapping. If happy about the script, then we can test it with the validate algorithm button, the green button, and then that will give a preview of the result. So it gives the first 10 examples, but it also gives a small overview of the total number of rows in your data set and how many the script could successfully map, and how many errors there are. So in the end, that should be all agree. The success should map total number of rows. And this is something that has to be done for each of the attributes and each of the data sets. So in the end, at least all the required fields should be green for all of these data sets, and then we can create the updated catalog by clicking on the Create the Integrated Dataset button. So then, as Mary already shows, that will create the catalog that allows searching for material, for instance, DNA 14 chromosome. So as a summary of what we do we need? So from you, we need your pseudonymized data. So it is the responsibility of each of the biobanks to make sure that there are no social security IDs or hospital IDs or governmental IDs for the patients in the data. We need a description of your data so we can build the mappings and the contact details of the person who's responsible for delivering your data. And then what we will do for you is set up the catalog to accept your data. So that is at least for the first time validate and upload your data, write together with you, write the mappings of your data to the standard model, and also provide you with the login details so that later on you can also update your sample data by yourself, as long as you can provide the data in exactly the same format as you provided the first time. You should be able to do that. If you're making changes in your source system and your export from there changes, then of course we have to work together to update that again. So it's a very brief summary of how we do this upload. So from each of the biobanks, we will generate an upload of your data as is, and then we convert it to the RD Connect standard data model by creating a mapping of your data to convert it. And yeah, so it's important that to notice that at the end of the RD Connect financing, we can't spend as much time on it to support you as we can now, unless we get other financing. So make use of the remaining time of the RD Connect project to get support in getting your data into the catalog. Then there's a few more links here also, the guides from the manual for the catalog and the form on the RD Connect website, some more information and movies for Mulgenus. So this presentation is also available on the RD Connect website together with the manuals, and we will also make a video of this presentation available. So are there any questions at this moment?